Okay, let's begin. So I want to go over this paper that has not been peer-reviewed yet called Potential Involvement of Monoamine Oxidase Activity in SARS-CoV-2 Infection and Delirium Onset. So you might be familiar with the monoamines because frequently they are psychedelics that are derived from, well, oftentimes they're derived from the tryptophan amino acid that gets turned into these monoamines. So let us look up what a monoamine oxidase is, and then maybe we can get some understanding on how it relates to psychedelics. So this is an example of the monoamine oxidases, and they will catalyze a reaction. So they are a family of enzymes that catalyze the oxidation of monoamines. And I shouldn't have said tryptophan. There are many types of monoamines, but these often are neurotransmitters and neuromodulators, and they contain one amino group connected to an aromatic ring by two carbon chains, such as ethyl-ethyl. So an example of this would be dopamine. So here we have this aromatic ring over here, and then it has this ethyl group, and then it has this monoamine, or this is an amine group, right? And so here we have norepinephrine, which is very similar to dopamine, but instead of having just ethyl groups here, we also have this alcohol group here as well. And here we have serotonin, which looks like tryptophan, right? So tryptophan would have a, what was the word? Well, we'll just keep on going. So this is also an aromatic ring with an ethyl group, and then here we have our amine. And there are countless different types of monoamines. And this, let me see, we have, let's look up DMT. So DMT, dimethyl trip, you know what, I'm gonna have to just look this up. DMT. So N,N-dimethyltryptamine is here. And so here we have our indole group. So if you look at this, this looks a little bit like serotonin, but it's not quite. So here we have this group, tryptamine, or this indole group. Here we have our ethyl group, and here we have our amine group. And this is N,N-dimethyltryptamine. So it has two methyl groups right here and right here. And so this is a psychedelic drug, and it has historically been prepared by various cultures for ritual purposes as an entheogen. And entheogens are things that generate sort of these supernatural experiences, at least within the minds of people who take them. So DMT is illegal in most countries. So let's see if I should go too much into detail. I don't think I should. Yeah, so just know that DMT is an example of a monoamine. And so the monoamine oxidases, let's look up monoamine oxidase again. So the monoamine oxidases are molecules that oxidize these monoamines. And they get oxidized in order to prepare them to be expelled from the body. So monoamines are important in the breakdown of the monoamines, which are ingested in food, and also serve to inactivate monoamine neurotransmitters. So these monoamine oxidases are destroying these neurotransmitters because you can't just have these neurotransmitters forever. They have to serve their function as a neurotransmitter, and then after they've served their function, they need to be degraded. And then there's a constant recycling of these neurotransmitters, and if this becomes dysregulated, you can end up with certain types of neurological pathologies. So because of the latter, they are involved with a number of psychiatric and neurological diseases, some of which can be treated with monoamine oxidase inhibitors, and these are called MAOIs, and these block the action of the monoamine oxidases. So, in humans, the mouse, there are two types, MAOA and MAOB. Both are found in neurons and the astroglia, or the astrocytes. And then some are found outside of the central nervous system. So MAOA is found in the liver, pulmonary vascular endothelium, the gastrointestinal tract, and the placenta. And the MAOB is mostly found in blood platelets. Okay, so let's understand what is going on here. So with that, let's go back to this pre-peer-reviewed paper, and I think I'll begin by just reading through it and then try to explain what could be happening in some of these patients who get COVID-19 and have an onset of delirium, or they feel like they are, what would the word be? Yeah, delirium would just be the best word, where things do not feel real, or they don't feel right, or they feel like they are being, that they're in this terrible nightmare or a dream while they are in the ICU. This seems to be a recurring thing. There seems to be neurological, psychiatric, psychedelic effects that COVID-19 is exhibiting or pushing onto the people who contract it. So there might be a mechanism by which this is happening through the production of monoamines, or the inability for these monoamines to be oxidized properly in the body. So let's begin. So severe acute respiratory syndrome coronavirus 2, also known as SARS-CoV-2, causes a range of extra respiratory signs and symptoms. One such manifestation is delirium, which is an acute confusional state, occurring in 60 to 70% of severe SARS-CoV-2 cases. So this isn't just something that very few people get. It seems that most people who have a severe form of SARS-CoV-2 seem to have some form of delirium. And now, granted, we could argue all day saying, well, if you are in a highly damaged state, you're probably not going to feel great anyway, and you're probably not going to be in the best state of mind while you are, one, not getting enough oxygen, or your body's really damaged, or you have these blood clots going on, so you probably won't be thinking correctly. Regardless, there might be more to it than just the damage. There might actually be some sort of chemical rationale to it as well. So delirium is also a common clinical syndrome following planned orthopedic surgery. This investigation initially explored the underlying role of metabolism in delirium susceptibility in this setting. So metabolomics profiles of cerebrospinal fluid. So the metabolomic profiles, these are, you take this cerebrospinal fluid and then you're running it through machines that can separate the chemicals or the metabolites from each other, and then it quantifies the metabolites, which are just all these chemicals that your body is producing from one to another. Okay, so and they also did it in the blood, and taken prior to the surgery they found significant concentration differences of several amino acids. And amino acids, these are the things that make up your proteins. The acylcarnitines, which let me get up an example of those. I wish Wikipedia was always the most recommended one. So here we have N-acetylcarnitine, which is technically also an amino acid, but it's not a proteogenic amino acid, or I should say it is a derivative of an amino acid, but not a proteogenic amino acid. And it's naturally produced in the body, although it is often taken as a dietary supplement. Acetylcarnitine is broken down in the blood by plasma esterases. So this is an ester group right here. So esterases will come in and break and remove that acyl group, and to which is used by the body to transport fatty acids into the mitochondria for breakdown. So just know that this is just another metabolite that is in different concentrations than the normal healthy individuals. So, and then the polyamines. And these were observed in delirium-prone patients. So the delirium-prone patients had different levels of these amino acids, acylcarnitines, and the polyamines than other patients. So the phenylethylamine, or the PEA concentrations in delirium-prone patients, was significantly lower in the cerebrospinal fluid than in the blood, while in age and gender-matched controls, the opposite was observed. So they did the adjusted p-value, and yeah, they were very, 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 different in concentrations. So the phenylethylamine, what is that? So this is like think of methamphetamine. So these are the amphetamines pretty much. So this is the phenyl group, or I should say, this is an aromatic ring, and then here we have the ethyl group, and here we have the amine, and that's why it's called the phenethylamine. It's not a phenol group, but it's a phen group. So continuing, PEA is metabolized by monoamine oxidase, or MAOB, which is a putative enzyme target for the treatment of Alzheimer's disease. And remember, this monoamine oxidase, this is found in the neurons, the glial cells, or the astrocytes, or astroglial cells, which are the nursing cells of the neurons. So they're not neurons themselves, but they are part of the nervous system in some way. And then this monoamine oxidase B is also found in the platelets, which are the blood clot-forming cells, or yeah, I think they are still technically called cells of the body, thrombocytes, so they are technically cells. So, a putative enzyme target for the treatment of Alzheimer's disease, Parkinson's disease, and depression. So, our computational structural comparison of MAOB and angiotensin converting enzyme, also known as ACE2, found high similarity. They ran a computational structural comparison. So, you can take, if you're doing bioinformatics, or if you're doing, if you have these computer programs that can model these enzymes or these protein structures, you can actually superimpose them on top of each other to see how similar they are in certain locations. And so they're saying that for the ACE2 receptor, the angiotensin converting enzyme 2, which is one of those surface or cell surface receptors that are that is in the cells that are susceptible to SARS-CoV-2, this receptor looks very similar to the MAOB, or the monoamine oxidase B enzyme as well. And so they found that there was high similarity, and specifically within the SARS-CoV-2 spike protein. So the MAOB structural alignment to ACE2 was 51% overall, but this was over 95% in the ACE2 spike protein binding region. So explicitly in the region of ACE2 where the spike protein of the coronavirus will bind, this area of MAOB shares 95% similarity with the ACE2 receptor. Okay? So this would tell to us, or at least would be a sign to us, that there is a likelihood, and a good likelihood, that the spike protein would be able to interact with the monoamine oxidase B enzyme as well as the spike protein using the same mechanism. So, or using the same receptor binding domain of the spike protein. So this is fascinating. Now, granted, this MAOB, I don't believe this is a cell surface protein. Let's look this up real quick. So it's an enzyme, and it belongs to the flavin monoamine oxidase family, and it is an enzyme located in the outer mitochondrial membrane. Oh, so it's in the outer mitochondrial membrane, so it's not on the surface of the cell. It's located within the mitochondria, which makes sense because oxidases are frequently found in the mitochondria, which are the powerhouses of the cell. They're the places that where all this oxygen is acting upon. So let me see. So it degrades dopamine. So monoamine oxidase B has a hydrophobic bipartite elongated cavity, and I don't think any of this stuff really matters for you. So here are the inhibitors of it. So I think we'll keep on going. Hopefully there isn't too much more that we're missing. So the structural alignment to the ACE2 was 51% overall, but this was over 95% in the ACE2 spike protein binding region. Thus, it is possible that the spike protein interacts with monoamine oxidase B on a molecular level. So, a previously published metabolomic data set of control subjects and patients with either mild or severe COVID-19 was then analyzed, and major concentration differences in some metabolites attributed to alter monoamine oxidase activity were detected. So they looked into the metabolomics, or the metabolome, or the different metabolites I should say, a profile of the different metabolites that people are producing. And in the SARS-CoV-2 or the COVID-19 patients, they had major concentration differences in some metabolites which are associated with MAO activity. Therefore, we hypothesize that the SARS-CoV-2 influences the MAOB activity, which is one potential cause for the many observed neurological and platelet-based complications of SARS-CoV-2 infection. So remember, MAOB is in the neurons, but it is also in the platelets. So if something is getting screwed up with monoamine oxidase B, it might also be screwing up the platelets. And I would also assume that I would have to look to see if the megakaryocytes also have monoamine oxidase activity. And in fact, let us just look this up right now just to see. So megakaryocytes, and the megakaryocytes, remember, these are the white blood cells that actually turn into the platelets. So they are, technically, I don't know if they are white blood cells, but they are a type of immune cell that ends up breaking up into the platelets. So in vitro growth of megakaryocytes, transcripts. Oh, no, I don't. Let's look up monoamine oxidase B. Platelet monoamine oxidase B and plasma beta something. So it looks like, I'm not saying anything about the megakaryocytes, but perhaps, yeah, it's saying here, I'm only seeing stuff for the platelets, not the megakaryocyte that it comes from. Regardless, we could continue. So this is one potential cause for the many observed neurological and platelet-based complications of SARS-CoV-2 infection, and so further research is required to establish what effect MAOB inhibitors might have on these pathways. There is no evidence at present to support the withholding of MAOB inhibitors. So let's say the spike protein is floating around in solution and it is able to interact with the monoamine oxidases. What does this mean? Well, this could be inhibiting the monoamine oxidases. They could be increasing the or I should say they could be activating the monoamine oxidases, making them more ready to cause catalysis. I mean, that would be unlikely, but that would be possible, right? So they could just be influencing monoamine oxidase activity, and granted, it could have preferential effects on certain types of monoamines versus another. So there could be a dysregulation of your monoamine metabolism based on differential inhibition or activation of this monoamine oxidase activity. So that is what could happen here then. Well, remember, your body is using these monoamines as neurotransmitters, right? So monoamine oxidase B, DMT. I wonder if, so inhibition of monoamine oxidase A is associated with let's look up DMT real quick. So yeah, when it comes to the tryptamines, the monoamine oxidases are put in there to prevent, so for instance, when people take ayahuasca, there's usually a monoamine oxidase inhibitor from some other plant, I don't remember which one, which allows the monoamines, or the DMT from the ayahuasca plant, this allows it to go from the stomach into the rest of the system because you have monoamine oxidases in the stomach as well. So MAOIs prevent oxidation of DMT in the digestive tract, which renders it biologically inert, and this allows it to be absorbed into the stomach and small intestine, allowing one to experience the effects of DMT by taking it orally, for example, in ayahuasca. So I don't know if this is with monoamine oxidase A or monoamine oxidase B, but you know, they both serve the same function, and they probably have differential preferences as to what monoamines they tend to degrade. So yeah, let's go back. So things that I want to say about this is this could be causing negative effects to people's mental states, probably long after the virus has already stopped multiplying. So even after the virus has stopped multiplying and exerting damage that way, these leftover spike proteins that are floating around in solution, or the leftover mRNA that is floating around in solution, could be creating more spike proteins even after the viruses are completely gone. Or I should say, even after the active virus is gone, there is still the possibility that these spike proteins are floating around and influencing neurological metabolic pathways, because they are either inhibitors or activators of the monoamine oxidase enzyme. So this is just something that I want you all to be aware of, is that this might be a future neurological complication that a large swaths of the population might experience. So frequently, I keep on hearing that people who contract COVID-19, they do not feel right, or things do not feel real, and this could, in fact, be a result of these monoamines getting dysregulated within the neurons. And later today, I'm going to be doing another video regarding how can these viruses even get into the brain in the first place. So there seems to be the possibility that it's being transported along axons, and so I want to explore that as well. So hopefully I can get that video out by the end of the day. But yeah, there's a lot that needs to be said here. And granted, this is not peer-reviewed. They're just saying that there is a possibility that this might be a direction that the research must go. And I need to also mention that these people, they're not in low-ranking positions in the medical community. So this Miroslava, she's a part of the National Research Council in Canada, I believe. And then this Emma Cunningham, she's trying to remember. I looked this up on Google once. So these people, they're at least in positions where I am willing to, what would the word be? They are risking their credentials, I should say, or they're risking their, what would the word be? Their face, right? They are putting their face on the line by coming out with this paper. Granted, they haven't made any really, really definitive statements. They're just saying that computationally there might be a interaction here. So I recommend that you all read the paper yourself, and the paper, I'll leave an article to the, or I should say, I will leave a reference to this article in the description down below. So if you want, this is the paper, and yeah, let me be honest, I didn't really fully read the paper yet, but and I'm sure that there's a lot that needs to be fixed because it isn't peer-reviewed yet, but hopefully this ends up panning out to be an effective treatment strategy for at least some of the psychological problems that people are experiencing with COVID-19. So with that, thanks for watching, and hopefully I will see you tomorrow.